Hello friends, today let us go on to the third topic of the series of immunology lecture, and this topic will be dealing with immunohematology. The term immunohematology can be broken up into the three words immuno, hem and logy. Immuno means immune system, hem means blood and logy means study. So immunohematology refers to the study of the blood group, blood transfusion, blood group antigen, antigen antibody reaction and the blood group disorders. As we can see, there are 14 blood group system has been discovered in man. Example, ABO blood group system has been discovered in 1900, MN blood group system was discovered in 1926, P blood group system was discovered in 1926 and RH blood group system was discovered in 1940. Here we can see that the blood group system and the year of their discoveries, various blood groups as well as their discovery years are given in this slide. When we talk about the ABO blood group system, ABO blood group was discovered by Landsteiner in 1900 and who was awarded with the Nobel Prize in 1932. They found two types of antigen in the human RBC, that is antigen A and antigen B. Similarly, there are two types of antibody that is present in the blood system, that is antibody A as well as antibody B. Based on the presence or absence of antigen and antibody, humans are classified into the four blood groups: blood group A, blood group B, blood group AB as well as blood group O. A person having the blood group A is having the antigen A and antibody B. Similarly, B group person contains antigen B and antibody A. A person having the blood group AB is having antigen A and B but has no antibodies regardingly. The fourth group is the O group, where the person contain no antigen but both antibodies A and antibody B are present in the system. In this figure you can see the antigens as well as the antigens which are present on the surface of the RBC. A person having the blood group A is having A antigen, a person with the blood group B is having the B antigen, a person with AB blood group is having both the antigen on the surface of RBC, and the person with the blood group O is not having any antigen type of this type, particularly A or B on the surface of the RBC. When we talk about the isoantibodies and isoantigens, the isoantibodies are belonging to the same species. Isoantibodies are the antibodies produced by one individual that reacts with the antigen of the other individual of the same species. Isoantigen of an individual which is capable of eliciting an immune response in an individual of the same species who are generally different and who do not possess the antigen is called the isoantigens. Here we can find the distribution of ABO blood group, and we can see that O blood group is very common blood group and the AB blood group is the rarest group. In this table you can compare the difference of percentages of various blood group person in different countries like India, America as well as Britain. When we talk about the blood transfusion, the transfer of blood from one person to another is called the blood transfusion. The person receiving the blood is called the recipient, the person donating the blood is called the donor. In the blood transfusion, the ABO grouping should be tested. When a recipient receives a wrong blood group, agglutination of the blood occurs in the body of the recipient and it leads to his or her death. This diagram clearly indicates how a person of different blood group can donate the blood to the another recipient. The donor, if the donor is O, it can donate the blood to almost all the blood groups, that is O, A, B as well as AB. A person with blood group A can donate the blood only to a person which is having the blood group A or AB. If a person is having the blood group B, it is able to donate the blood to B as well as AB person. And on the other hand, a person with the AB blood group can donate the blood only to AB. So here we can say that O is the universal donor as well as the AB is the universal the recipient of the blood group from various people. Landsteiner and Weiner in 1940 discovered the existence of a special type of antigen in the RBC of rhesus monkey, rhesus factor or the RH factor as it is known. The RH factor is also present in the RBC of some of the human being. So it can be classified in the two groups, that is RH positive as well as RH negative. Rhesus factor is an inherent protein found on the surface of red blood cells. If your blood has the protein, you are RH positive. If your blood lacks the protein, you are Rh negative. Rh positive is the most common blood type. It is also called the D antigen. Here is the percentage of presence or absence of Rh factor in different countries, and we find that Rh positive persons are more frequent in many places. And the Rh antigen is not having any natural antibody for it. How Rh system and blastocyst fetalis is related, here we can discuss. During pregnancy, if the mother is Rh negative and if the father is Rh positive, the child may be Rh positive. Rh positive red blood cells may begin leaking crossing the placenta into the mother's cardiovascular system, since placenta tissue normally breaks down before and at the birth. The mother produces anti-Rh antibodies which may cross the placenta and destroy child's red blood cells during a subsequent pregnancy. This diagram indicates the various stages of the erythroblastosis fetalis. The medical applications of blood group are also very important, because blood group is having direct role in the blood transfusion, hemolytic diseases, transplantation, solving disputed parentage and detection of culprits. When we talk about transplantation, the success of skin graft and organ transplantation depends on the compatibility of the blood group. When we say about the detection of the culprit, in case of murder, the culprits are identified if the blood stains are left. In case of disputed parentage, the disputes regarding the parentage of child, the blood group analysis can solve the problem. Now, this is a small video which will help you to understand better how the blood group testing takes place. In this video, you are going to find that there is availability of the kit in which the monoclonal antibodies for different antibodies are present, that is antibody A, antibody B as well as antibody D, that is meant for the rhesus factor. The samples are taken into the slide and then the antibodies are mixed with them, and when we find the clumping, we indicate that the test is positive for that particular antigen present in the blood group surface. So, here I would like to thank you, and I would like to suggest that give the gift of life and donate blood as you can. Thank you.